Let's talk about gram staining. Gram staining is used to identify different types of bacteria. And this is basically a lab procedure where you're going to use different inks and dyes to dye bacteria to identify what type of bacteria it is. When gram staining, there are two main types. Gram-positive bacteria turns purple in a gram stain. Gram-positive bacteria have a thicker wall, a single membrane, and a molecule known as peptidoglycan on the outside. This is what the cell wall looks like for gram-positive bacteria. You can see the peptidoglycan, which is a molecule found on the outside of the cell. This is the substance that turns purple when a dye is applied. Gram-negative bacteria, on the other hand, turns pink. That's because there is a thinner cell wall in these bacteria and there's actually a double membrane. The peptidoglycan in these bacteria is sandwiched in between the membranes. So you can see the two membranes in gram-negative bacteria and the peptidoglycan being located between them. So basically, gram staining is just another way that we can identify types of bacteria using different lab procedures. Because when we find bacteria, say on a lab bench, and we don't know what it is, we have to identify it. And it's not that easy to identify bacteria because it's so small. So we use these gram staining techniques to kind of get us started into identifying a strain of bacteria.